Hello, my name is Alexis Guerra. I am a first-year PhD student in the Environmental Health Sciences program at the University of California, Irvine. I work in Dr. Sunny Zhang's lab, where we are using methods to detect SARS-CoV-2 in our coastal environments. Now, I think we can all agree that we humans rely heavily on resources from the natural environment around us. We also depend on these resources to be safe for us to use, since we are interconnected with this environment. That being said, our actions can also potentially inflict damage on the environment around us. In the Zhang lab, we study how human activities can spread pathogens like viruses and bacteria throughout the environment, which can in turn harm human health. An example of this kind of human health issue in the environment is the very same pathogen that is keeping us all at home today, SARS-CoV-2. Shedding of this virus from human waste has been shown to contaminate the environment, including in waters and sediments that we are potentially exposed to. So in our lab, we are testing methods to detect traces of SARS-CoV-2 by sampling coastal seawater and beach sand from a beach of interest. We filter the samples and extract any viral RNA of SARS-CoV-2 in the samples. And then we convert this RNA to DNA to perform droplet digital polymerase chain reaction, or DPCR. Then we use the results from this to calculate the levels of SARS-CoV-2 in the samples. Today, I'll explain DPCR a little more in detail. To understand DPCR, we must first recognize that traditional PCR is a technique that makes many copies of DNA that we are interested in looking at. This is a method commonly used in crime investigations. If detectives find a drop of blood at a crime scene, they can collect that blood and use PCR to make enough DNA for them to decide to whom does the blood belong to. However, since we work with beach water and sand samples though, you can imagine that there are millions of microbes that can obscure this tiny specific DNA that we are interested in. So we have chosen to use DPCR, which uses fluorescence and droplets to better detect minuscule amounts of DNA in samples such as these environmental samples. Now here's how it works. We put the viral DNA into this device that's kind of like a bubble machine because it breaks the DNA into thousands of tiny droplets. Then we would actually run the PCR cycles, which is when the viral DNA is being copied. Now let's imagine that SARS-CoV-2 is represented by this green band, our target of interest in each of these droplets, and the blue bands are the DNA from the rest of the microbes. All the DNA will be separated into each of these droplets with a different chance of each droplet actually holding our target DNA within. As the DNA is being copied over and over, the droplets that do have the target DNA of interest will show a bright green light, and we can use this to measure the amount of target DNA or SARS-CoV-2 present in the sample. Based on these concepts of DPCR, we should be able to detect trace amounts of SARS-CoV-2, if there is any, in the water and sand samples. Now, if we were to detect SARS-CoV-2 in these samples, these results could reveal how much human waste is flowing into our coastal ecosystems. What could this mean for human health or the health of the marine life in the coastal ecosystem? This would also contribute to another study in the Zhang lab that is using DPCR to trace SARS-CoV-2 in sewage water and to estimate the amount of cases in human communities based on those levels. And with that, thank you so much for listening. I hope that you and your loved ones are safe and healthy.